This program is a presentation of UCTV for educational and non-commercial use only.
Good evening. My name is Nancy Asher, and I'm the chair of the Department of Surgery, and want to welcome you to this mini-med session on transplantation. I think many of you were here last week, and you heard the beginning of our series, and this is a continuation of the series. It's a little bit more advanced today. You're going to hear from two of our fabulous faculty. Dr. Sang-Mo Kang is an associate professor in the Department of Surgery. He did his undergraduate training at Cornell and then received his medical degree at Harvard and joined us first as a resident, I think, in 1992, and now has joined our faculty. He's grown up now, has joined our faculty, and he actually is the director of the intestinal transplant program, but his interest in the laboratory is how to make people not recognize tissue as foreign, so how to induce tolerance, and he's also interested in how to build new organs so that our terrible wait for solid organ transplant can be avoided and people can get treated earlier. Sang-Mo, why don't you begin?
Well, thanks, Nancy. Thank you all for showing up. I can assure you that in medical school we never showed up in the evenings, except when we were on the rotations. But I'm just going to give you a little bit of background today about transplantation, just because I don't know how many people were here last week, and it's probably a good idea just to give a little bit of very brief background. We don't have that much time. And we'll talk about the two major problems that face us today in transplantation and how stem cells and organ engineering might be able to address those issues. So in terms of the history of transplantation, it really didn't start until Dr. Alexis Carrel developed a way of hooking together blood vessels. If you think about it, an organ transplant really depends on blood flow in and out, and other tubes to be hooked up for bile or for the urine. So this was a critical element, and Dr. Carrel actually observed that organs would last for a short period of time, but then eventually get rejected or stop working. He didn't exactly know why. Dr. Medawar was able to figure out that the immune system was actually rejecting these organs, and began to explain the mechanisms that related to the rejection, and he was awarded the Nobel Prize in 1960. Dr. Joe Murray performed the first successful renal transplant and was instrumental in developing new immunosuppressive drugs that were critical to graft survival, and he was awarded the Nobel Prize in Medicine in 1990. And the immunosuppressive drugs are really key, and Dr. Stock will be talking a little bit about how problematic rejection can be in our patients even today. The history of transplantation goes back a long way at UCSF as well. Dr. John Najarian was the head of the transplant program from 1963 to 1966, and actually went to Minnesota to head the department there and trained Dr. Asher as well as Dr. Stock. Dr. Volkert Belzer was also at UCSF and then went to University of Wisconsin and trained a huge number of people there, lots of leaders in transplantation. And he was instrumental in developing organ preservation techniques that really allow us to do the transplants that we do today, transporting organs from one place to another. Dr. Starzl was a true pioneer in liver transplantation and performed the first human liver transplant in 1963. And back then, there was a tremendous amount of hurdles and very bad outcomes at the beginning. But he was able to stick it out and really turn liver transplant into what it is today, a very well accepted and effective treatment for end-stage liver disease. He was instrumental in developing cyclosporin and tacrolimus, which are very important immunosuppressives today. So today, transplantation, after all that, is the optimal therapy for a variety of end organ diseases. And 16,000 kidney transplants were performed in 2007 in the U.S., so fairly large number. And overall patient and graft survival rates have improved dramatically since the early days. But, well, and you guys may or may not know about these high-profile recipients. This is Alonzo Mourning. He got a kidney transplant and then won a championship a year later. Sean Elliott played for the San Antonio Spurs and also won a championship the year that he came back. So I'm not sure if that's a common theme or not. Maybe I can convince one of the Dallas players to get a kidney transplant. And then Chris Klug actually won the bronze medal in snowboarding 2 years after a liver transplant. And he was actually transplanted by one of our Dr. Asher and Dr. Stock's trainees at the University of Colorado. And then at UCSF, we performed over 10,000 transplants in our 45-year history, and the most kidney transplants of any center in the world. 2010, we performed over 300 kidneys and 140 livers, 21 pancreases, so quite a large volume. So what's the problem? Well, organs don't fall off trucks, livers don't fall off trucks, and kidneys don't grow on trees. That's one of the main problems. And so what you see is that the number of patients on the kidney transplant waiting list is increasing dramatically, and the number of deaths of patients who are on the waiting list is also increasing. If you look at liver transplantation, again, the number of patients on the liver transplant waiting list is going up rapidly, and the number of deaths on the waiting list is also increasing, as you would expect. Because the total number of donors over that same time period hasn't really kept up. There's been a slight increase in cadaveric donors, and we've been much more aggressive about using more marginal donors, but the number of living donors has actually gone down slightly. So that translates to longer waiting times. For a kidney, for a cadaver kidney, is 6 to 8 years in our area. We have over 3,000 patients on our waiting list alone, and there's over 100,000 patients waiting in the U.S. So it's a huge problem. The other big problem that we still face is that when you get a transplant, you literally have to take a handful of pills every single day, and they can obviously have many side effects and cause lots of problems. So this is still a problem today. So when we see patients in the clinic who are coming in for a transplantation eval, a question that we'll often get is, after we describe all the problems of waiting and the medications, they say, well, what about stem cells? Can't you just make me an organ? How far away is that? And this is actually a fairly common question. And you wonder where they get those ideas. And if you do a Google search for stem cells, you see all kinds of stuff: fetal stem cell treatment, all kinds of stuff coming up as if it's a current therapy. There's been companies built on the idea of turning stem cells into all sorts of different types of tissue that could treat diabetes, spinal cord injury, heart failure, et cetera, et cetera. So there's a lot of hype out there about what stem cells can do. So what I'd like to do is first talk about the history of stem cells, define what a stem cell is, talk about the types of stem cells that have been characterized, what has really been achieved with stem cells to date, and then talk about the problems and challenges of stem cell therapy, because you don't really hear about that when people talk about science on the news, and then some possible solutions to these problems. So a brief history of stem cells. In 1981, embryonic stem cells were isolated in mice at UCSF as well as at the University of Cambridge. In 1995, embryonic stem cells were derived from monkeys, so now moving up to primates. In 1998, human cells were isolated at University of Wisconsin and at Johns Hopkins, and these were derived from aborted fetuses, which raised the whole controversy that still rages even today. The NIH in 2000 issued guidelines allowing federal funding of stem cell research, and very shortly thereafter, President Bush placed a hold on funding. And that prompted California in 2004 to pump $3 billion into stem cell research, and UCSF has been the beneficiary of that, as well as all the other campuses around California. 2006, the Senate passed a bill to expand federal research funding for stem cells, and shortly thereafter, President Bush vetoed it. There's still, even though Obama tried to turn that over recently, there's still a lot of court battles going on, so it's very difficult to get funding for human stem cell research. Well, what is a stem cell? So the term stem cell actually was coined in 1868 to describe the first cell that ever was, that led to all organisms. At some point, there had to have been one cell, so that was the original stem cell. In 1877, he extended this definition to a fertilized egg that could give rise to all cells of an organism. The modern definition that we use today is a cell that can self-renew and give rise to differentiated cells. So it's a little bit more of a general term. This is a schematic of how you would isolate a stem cell. Basically, a zygote is what we call totipotent, or it can make every single type of cell, obviously. This is a fertilized egg. After a few days of development, you get this blastocyst with this inner cell mass. If you take these cells out of the blastocyst and put them in a culture dish, you have what's called embryonic stem cells, and that's what a lot of stem cell research is based upon these days. These cells are pluripotent, meaning that they can make all the different cell types in the body. And so they can make nerve cells, bone marrow cells, liver cells, skin cells, et cetera, et cetera. So it can really reconstitute an entire animal or person. What's been achieved with stem cells today? Well, bone marrow transplants have been around for quite some time. These are not embryonic stem cells, but bone marrow transplants have been performed for decades, and is a very useful approach to curing many diseases. And so that's really the biggest success of stem cell research, if you want to have a broad definition. Now, in terms of embryonic stem cells, what's been done? Well, primarily, so far we've been able to generate different types of cells in vitro or in a test tube. So heart muscle cells, bone marrow cells, liver cells, neurons, muscle cells, and endocrine cells like the beta cells that make insulin. It's also allowed us to actually target certain genes and either knock them out or add genes to an entire animal. We don't have really time to get into that today. It's been shown to repair some tissues in experimental models, and we'll go into that a little bit. It's also been very useful for cloning animals. And you guys have probably heard about Dolly, but the first sheep that was cloned. And so what you have is an adult tissue donor. And basically you grow these cells, the cells from this adult in a dish. You remove the nucleus, or the part of the cell that has all the DNA and all the genetic information, and you put it into an egg, essentially. So you take a egg from from another donor, you suck out the nucleus from that egg, and you put this adult nucleus in, and you let that develop into an embryo. It makes it sound a lot easier than it actually is. It's not that easy, but that's the general schematic. And then you end up with Dolly. And these are all the different types of cloned animals that have been generated so far. This is a ferret, actually. I don't know who thought of that. Sheep, donkeys, deer, ponies, dogs, pigs, free martin cattle, gar, which I understand is a buffalo-like animal. But all sorts of animals have been cloned. And of course, human cloning has been banned. There was actually a company that was set up to clone your pets. So this is a company called Genetic Savings and Clone. And it's no longer in business, but this was a Bay Area firm that actually was able to clone cats. They weren't able to do dogs, but they were able to do cats, and they were obviously going out of business because they had a cat cloning price reduction. But I can imagine that that's going to come back again someday. And then there are unintended consequences of cloning. So according to the president, stem cell cloning can have disastrous consequences. And then there's some other, this is actually a movie with Scarlett Johansson and Ewan McGregor. It's actually a pretty good movie, but the concept is that there's a whole colony of clones that are being raised. They don't know that they're clones. They're living this idyllic life, and then these guys escape and figure it all out. But I do like the idea of cloning Scarlett Johansson. So what is there evidence for tissue repair in vivo of these stem cells? Well, there's animal models that have been developed of Parkinson's, amyotrophic lateral sclerosis, liver injury models, spinal cord injury, and heart injury models or heart attack models. And in those models, you can show that if you inject certain types of stem cells, that there may be some improvement in parameters. Now, when this was tried in humans, this was actually tried in humans where they injected some bone marrow stem cells into hearts after a heart attack, and they really found no benefit. So we always have to be a little bit careful about extrapolating animal studies to humans, but certainly there's a lot of animal data suggesting that there's a benefit of stem cells. What are their potential problems in stem cells? Well, it turns out that if you have a true stem cell, an embryonic stem cell, and you inject it into an animal, that they'll actually turn into tumors that are called teratomas. These are tumors that will make all sorts of different tissue in the tumor. There's also the possibility for genetic aberrations because you're growing cells in a test tube, and every time it divides, there's a chance for chromosomal instability. There's some question as to telomeres. These get shorter and shorter as the cells divide over and over again, and eventually, when they get short enough, the cell dies. So it turned out that Dolly had telomeres of a 6-year-old at birth, and so that's a concern. And then there's immunologic barriers. So it turns out that stem cells aren't immune privileged or won't evade the immune system. If the stem cells are from a different person or different animal, if you're doing animal studies, they will incite an immune response and be rejected also. So that's another consideration. So this is an example of the teratomas that can form. So these were stem cells that were injected into the heart, and you can see that after a period of time, they're forming tumors on the heart. This is the rest of the heart there. Not a good thing, obviously. And then if you actually, this is a method to actually track the growth and spread of these tumor cells. So if you look, this is at day 2, day 4, it's getting bigger. Now it's starting to spread, and you can see this tumor spreading throughout the body. So that's obviously not what you would want to have happen. And this is something that's not widely known in sort of the general community, that these cells can be problematic. this is just a slide showing that, that the stem cells can actually be rejected as well. Now, so what are the future needs? Now, I put this slide up just to demonstrate how fast this field is moving. So this was a slide I made in 2007 about future needs in stem cell therapy. So one was that we needed autologous stem cells. You want to have your own stem cell, whether it be for therapeutic cloning, although it wasn't really sure whether you could do this in primates. And then, or the other concept would be to take an adult cell and turn it into a stem cell by de-differentiating it or going backwards in differentiation. And this would eliminate a lot of ethical barriers because you wouldn't need stem cells from aborted fetuses. You could get stem cells from any of us. And then the other need would be to manipulate stem cells to decrease immunogenicity if we can't use autologous stem cells. Well, very shortly thereafter, it was shown that therapeutic cloning could be performed in primates. And in a really big development, it was shown that you could take adult cells and turn them into stem cells, and we'll go through that a little bit. So this is actually work that was published by a scientist named Yamanaka, and he did a lot of the work in Japan, but is also working at UCSF over at Mission Bay. And the concept was this. So basically they took skin cells or fibroblasts from an adult mouse, and they put in four genes that they thought might turn on the stem cell phenotype or make them look like stem cells. And what they were able to show was that indeed you could make cells that kind of look like embryonic stem cells. And when they injected these stem cells into the blastocyst that I showed you earlier, these cells would mix in with these other cells and then make chimeric mice. So they had cells of the original embryo and also cells that were developed from these cells that were injected. And then you could breed these mice and develop what we call an IPS or adult stem cell derived mouse. So they were able to actually successfully turn an adult cell and get progeny from that subsequently. So this was a huge development, and this showed that you could actually make stem cells from any adult. And these cells were able to make intestinal cells, muscle cells, skin cells, cartilage. I'm sure a lot of us could use that. We don't need added fat tissue. And then some brain, nerve tissue, which we could probably use also. So pretty powerful stuff. And then this was sort of a home run type of experiment. So this was an experiment where they wanted to treat an experimental model of sickle cell anemia using embryonic-derived stem cells. So what they did was they took a mouse that had it was a mouse model of sickle cell anemia. They took the cells out to make the stem cells from this adult mouse. Then they actually put a gene in to fix the defect that causes sickle cell anemia, made bone marrow cells out of these embryonic stem cells, and then did a bone marrow transplant into that same mouse that was the donor for the stem cells. And they were able to show that indeed they could fix sickle cell anemia in that same animal from embryonic stem cells. So it's pretty remarkable set of experiments. This was only recently published in 2007. Our own faculty in our department, Holger Willenbring, was able to was published in 2010 that these adult stem cell derived cells could actually repopulate a liver that would otherwise fail. So this was also a very nice demonstration that these adult derived stem cells could be useful for therapy. So future directions, we really need to establish and improve the safety of gene modifications for differentiation of adult cells into stem cells. And you know, a lot of people are working on non-genetic methods because genetic methods themselves can be problematic. We need to refine ways of taking a stem cell and turning it into exactly the type of stem cell that we want. We have to figure out ways to eliminate undifferentiated stem cells that we know can turn into tumors. The other thing is we need to figure out how to deliver these cells. You know, what's the best way? Do we inject them? Do we put them into the blood vessels? Do we inject them directly into the organ? There's a lot of things that we don't know, and this is part of where we get involved as well as surgeons. We do have a few stem cell trials in humans at UCSF. One is for a disease called Pelizaeus-Merzbacher disease, PMD. It's a very rare disease. And basically, it's manifest by seizures and delayed motor milestones. They have trouble moving and sometimes even breathing. And the basic defect is that when you have this disease, you don't have myelin, which is sort of the insulation that allows electrical signals to go through your nerves. So nerves transmit electrical signals, and if you don't have myelin, it doesn't work very well. This is like an insulator. And experimental studies were able to show that if you injected neural stem cells into experimental mice, that this would actually improve their muscle tone and their behavior. And this is a universally fatal disease, and there's probably about 50 patients in the world with this disease, thankfully. So basically, and they all tend to be males, and so anywhere from 6 months to 5 months of age. We actually take allogeneic stem cells. These are from different donors, and they're differentiated into neural stem cells and actually injected directly into the brain of these patients. Because they're from not from themselves, we do use immunosuppression with tacrolimus and mycophenolate, which is what we use in solid organ transplantation, and we keep that going for 9 months. And it's been pretty interesting so far. We also have some work going on with beta cell replacement. Dr. Stock is actually the PI, and Chi-Zi Tang is the co-PI, a PhD researcher in transplantation. And basically, they're trying to develop a method of developing islets from embryonic stem cells, because doing islet transplants for diabetes is great, and it works in a good percentage of patients, but there just aren't enough islets to go around. So if we could make islets, that would be really an amazing achievement. And so we're studying the immunologic response and also just how to best deliver these ES cell-derived islets. But it's one thing to inject cells or even islets, which are very tiny little balls of cells. You know, this is a ball of cells. How do you go from that to this? This is a model of a kidney. This is a much more complex thing. You know, you have many different types of cells. You have blood vessels. You got the urinary collecting system. I mean, how are you going to make that in a test tube? That's really the conundrum. And it'd be nice to make one of these as well in a test tube. So a really amazing achievement was made by Harold Ott and Doris Taylor at the University of Minnesota. And this was published in 2008. What they found was you could take a heart, this happened to be a rat heart, and basically perfuse them with detergent. And that would remove the cells. So you can see this is the heart before perfusion. It looks, you know, red because of all the muscle cells. And as you perfuse these hearts with detergent called SDS, it actually turns into what's called a ghost because all the actual cells have been removed. And when you look under the microscope, you can see that the cells are gone, but all the what we call the interstitial tissue remains. And this is some other pictures of this, of the skeleton that's left over. So All the cells are gone, but all the structural elements remain in this tissue. So the idea then was that, well, maybe if you take this organ, remove all the cells, maybe you can put new cells in. So you put cells in that you derive from the stem cells, or maybe young muscle cells, and try to make a new organ. And in fact, they did that. They after decellularization of the heart, they actually injected new heart cells in, and were actually able to make something that actually contracted. This is an EKG signal showing that there's contraction and electrical activity within this reconstituted heart. Let me see if I can get this movie to work. So this is an example of a heart that's been reconstituted after getting rid of all the cells. You put new cells in, and the heart is now beating. Pretty amazing stuff. Now, this heart's not beating very well. This is probably the kind of heart that you'd want to replace. But as a proof of concept, it was very impressive. You can also do this with kidneys in terms of decellularizing them. So this is, again, these are mouse kidneys, actually, that we've been working with in our lab. And you can basically take these kidneys and perfuse them and get these ghosts as well. You can do that with liver. So here's a liver that starts out, this is a normal liver, and then you start perfusing. You can see how the cells just start going away, and you're left with this beautiful ghost with all the little blood vessels still intact. And this is just another example. This is actually a paper from a group from Wake Forest, but basically they're showing the same thing with the liver, where they decellularize the liver. They have some beautiful pictures of the ultrastructural elements, these little blood vessels, and then portal triads, where you have a vein and an artery and a bile duct, and that bile duct still is intact. So these are the important elements in a liver, not just the liver cells themselves. There are alternate approaches that you may or may not have heard about in the news. One is that a lot of people ask me about is this printing of organs using inkjet technology. So basically what they do is they take some extracellular matrix and print in a three-dimensional fashion with the extracellular matrix and some cells, and they can actually build bladder wall and certain structural elements. And the idea would be that maybe someday they can make an organ out of it, although that's going to be very, very complicated. The other is to utilize silicon wafer technology, believe it or not. You can actually pattern silicon chips, essentially, and put cells on them, and that's work that's being done by one of our UCSF researchers, bioengineer named Shuvo Roy. He's also at Mission Bay. But he basically took silicon wafers, this is what they look like under the microscope, and loaded kidney cells onto these wafers, and were able to show that the cells actually orient themselves in a certain way so that they could potentially filter blood and then have the output go in the right direction. And his concept is that you would put a whole bunch of these things together and put it into a device like this. You can see how it says UCSF on it. Blood supply coming in, blood going out, and then a urine going to the bladder. So, this is a very futuristic concept, as you can probably tell from the illustration, and we're many years away. But these are the kind of approaches that people are thinking about, and how do we take cells and turn them into an organ? 'Cause it's one thing to treat with cells, it's a whole other thing to create this very complex organ. So no truly functional organs have yet been created. And then, if you think about it, it's one thing, the heart is actually one of the most simple organs, in that all it does is kind of pump. And so if you look at the other organs that we have, like the liver and the kidney, there's many different cell types, and they're all doing a lot of different things. And so really recellularizing organs with multiple cell types and micro niches will be very complex, I think. And so we're going to have to get through a lot of hurdles before that happens. The other thing is if you use silicon or other types of hardware, the human body really doesn't like hardware, and there's all sorts of problems that go along with that. So the bottom line is there's a long way to go. So we've gone over the history and background of organ transplant, two of the major problems, and how stem cells and organ engineering might impact those problems. And so I think the take-home message is that it is an exciting, fast-moving, promising area of research, but there are many hurdles that remain before it's really going to become reality for people who need organ replacement. That is the trillion-dollar question, right? I mean, one of the miracles of life really is how a single cell turns into, every kind of animal there is. Nobody truly understands how that happens. You can't make it happen in a test tube. It happens, in the uterus, and many people are actively trying to figure out how that happens. But when you take have an artificial situation in the test tube, it's much more difficult to control that. Nature has much more complex ways of figuring out where things are supposed to go, where the liver is supposed to go, the kidney. I mean, if you think about how complex that is, it's just an amazing thing. And so we're not that smart yet.
So I guess my understanding is sort of that I don't understand how Jolly. So they actually grew Jolly in sort of
Yeah. So what?
artificial environments. It wasn't really like manufactured.
It's like in vitro fertilization. So what they did was they took an egg, they sucked out the nucleus from that egg, they put a new nucleus in that carried the genetic information, let that develop for just a little bit in the test tube, but then all of the rest was done in a surrogate sheep. I didn't have time to get into that in detail, so thanks for bringing that up. The question is whether different organs have different success rates in terms of decellularization and then recellularization, and the answer is yes. As I was alluding to, the heart is actually the simplest thing. It's just a pump, and you can replace the heart with a pump, and that's been in the news all over the place. People can actually go home and live with a pump. So that's actually the simplest of all the organs, and even then, it's not really a functioning heart. There has been some work recellularizing a lung, for example, and showing that it has some function, but we're talking about 1 hour, 2 hours in an animal. So these are really in its infancy. The liver has also been kind of reconstituted, and there was some evidence of function, but it's really not convincing at all. So I think this is really within the past 2-3 years that this stuff has come on the scene, and so it's the whole field is in its infancy. But having said that, the early successes are sort of the low-hanging fruit, and it's going to be much more difficult to get to the very top. So the question is whether where cord cells are fit into the paradigm of stem cell treatment. And the answer is that cord blood stem cells are really just bone marrow stem cells. It's a very good source of bone marrow stem cells, but it's not a source of any other type of stem cell. So it's great if you want to store it in the case you need it in the future for a bone marrow transplant, but it's really not useful for these other types, regenerating other types of tissues. Yeah, that's a great question. So the question was about cartilage repair and simpler types of tissues that might be beneficial, especially as the population ages. I don't mean that in a bad way, but anyway. So microfracture, that's called microfracture surgery, where you drill into the bone. Like many basketball players, for example, have had microfracture surgery to try to regenerate some cartilage. And it turns out when you drill into the bone that there are some bone marrow stem cells, and somehow nobody really truly understands it. There's some cartilage that develops. It's not very effective. I mean, it works when you have small defects, but when you have a, true osteoarthritis, when you're 65 and you're, bone on bone, that's really difficult. So there is a need for that type of work, and I think that's really where the promise is for the short term. These complex organs are going to be incredibly difficult, so it's really about cartilage, bone, bone marrow cells. Those are the things where we're getting simple solutions that I think there's going to be much more advances, but there's a lot of work going on in cartilage repair and regeneration. Basically, you can think of it as bioprosthetics, real tissue that you can use instead of plastic tubes and things like that, because the body really doesn't like plastic or metal or anything like that. So the question was about getting organ transplants in other countries, and that's a whole nother bag of worms, but it is a very interesting topic, which actually came to light for the general public around the time of the Beijing Olympics, because China was really a hub for what we call transplant tourism, where people would go to China and get a kidney or a liver from prisoners who had been executed. And obviously that was quite controversial. They have scaled it back because of international pressure, especially around the time of the Olympics, because they didn't want to look bad. But it still occurs. It still occurs, and it's not just China, but it's other countries, India, Philippines. There's a whole slew of countries where it happens, and it's really an ethical disaster. If you think, some people call it transplant cannibalism. On the other hand, if you're the patient and you're looking at an 8-year wait list for a kidney, and you know that every year on the list you have a 5 to 10% chance of dying while waiting for a kidney transplant, and you have a couple of kids, and it's not quite so absolute. So I think it's a very difficult issue. I think it's important to remove that temptation. I don't think you can really blame the patients who are in that situation, because it's human to want to go and get better. So it's a very difficult topic, and it's something that we're going to be faced with more and more, I think, in the future. Yeah, and there's a potential of stigmatizing donation as if and making donation more of a lower middle class type of issue, just like the military is now. I mean, we send our poor kids off to get killed, but there's another type of transplant tourism that's within the United States. You saw it with Steve Jobs, and so there's many disparities amongst different regions. So our most large metropolitan areas have very long waiting lists, or you need to get really, really sick before you get a liver. And when you go to Tennessee or Florida or wherever, you can get transplanted right away. It's incredibly unfair, and yet the economics and the sort of the interests of different localities keeps this very disparate, unequal system alive. So there's a lot of ethical issues, but that could be like a whole series of seminars and discussions, but very interesting.
Go ahead. It's a very important question.
I think the oldest...
Can you repeat the question?
Sure. Oh, the question is there a limit to the age of the donors that can be used? And the answer is that the older the donor organs are, the less well they work. Now, that doesn't mean that everybody who's 80, for example, is not the same. There's really healthy 80-year-olds, and there's really unhealthy 80-year-olds. And so we have used donors that were over the age of 80. I think 82 is probably the record.
We had a living donor who was 79
Yeah.
who gave to her husband who was 78.
So I used a liver that was from an 82-year-old donor and worked great, but most of them we turned down. So I would say 90s it would be really pushing the envelope, but who knows?
So,